Okay, let's unpack this. Flesh-eating maggots. Seriously, the New World screwworm fly. Just think about that for a second. Larvae that burrows into living tissue. It's pretty gruesome stuff, right? What could possibly be the hidden mechanisms driving such horrific parasitic behavior?
Well, what's really fascinating here, maybe a bit unexpected, is that while our first thought might jump to, purely genetic reasons for something so extreme,
Hmm.
the science is increasingly pointing towards something more dynamic, epigenetics.
Epigenetics, okay.
We're talking about heritable changes in how genes are expressed,
Hmm.
but, and this is key, without any alteration to the actual DNA sequence itself.
Right, so the underlying code stays the same, but how it's read changes.
Exactly.
So the environment could actually be like talking to these flies at a molecular level, switching genes on or off that lead to this parasitism.
That's the idea. The sources we looked at really dive into this, exploring how these epigenetic mechanisms might be the key, or at least part of the key, to understanding this in screwworms and, similar species.
Okay, and you mentioned locusts too. How do they fit in?
Ah, well, they were a fantastic parallel. Think about locusts. You have this solitary, pretty harmless grasshopper that, under certain conditions, blam, it transforms into a swarming, destructive locust. Our sources highlight that epigenetics seems to be playing a really significant role in that dramatic switch as well. It's another case of environment flipping a behavioral switch.
Right, that incredible transformation from, yeah, harmless to, well, a plague, basically. So the mission for this deep dive, then, is to try and understand how the environment might be interacting with the genes of these insect flies. Locusts leading to these really dramatic changes in behavior. Let's maybe start by getting a clearer picture of what exactly epigenetics involves. What are we talking about molecularly?
Okay. At its core, epigenetics describes the processes that control which genes are active and which are silent, all without changing the DNA code itself, like we said. The main players here are things like DNA methylation. That often acts to silence genes by adding a little chemical tag, a methyl group, directly onto the DNA.
Okay, methylation equals silencing, generally.
Often, yeah. Then there are histone modifications. Histones are the proteins that DNA wraps around, like spools for thread.
Right.
And adding chemical tags to those proteins can affect how tightly or loosely the DNA is wound. That changes how accessible the DNA is for the machinery that reads the genes.
So looser means easier to read, tighter means harder.
Pretty much. And finally, there are non-coding RNAs. These are small RNA molecules that don't code for proteins themselves, but can regulate gene activity after the DNA has been transcribed into RNA. Sort of like fine-tuning.
Okay, so if our DNA is the blueprint, epigenetics is like the foreman, maybe the annotations on the blueprint, deciding which parts get built and when and how intensely.
It's a really helpful analogy, yeah. And to see this working in insects, the sources bring up the classic, really remarkable example of honeybees. You have genetically identical female larvae, right? But they can develop into either queens or worker bees, and they look and act totally different.
Totally different. And that massive difference, that fork in the road, is determined just by what they eat as larvae.
Exactly, just by diet. The larvae that are fed royal jelly are destined to become queens. And this royal jelly, it contains compounds that actually inhibit specific epigenetic enzymes.
Oh, interesting. Which ones?
Specifically, a DNA methyltransferase the enzyme that adds those methyl tags, and a histone decetylase, which removes acetyl tags from histones.
Okay, so royal jelly blocks the enzymes that usually silence or tighten things up.
That's right. By inhibiting these enzymes, the royal jelly leads to lower DNA methylation and higher histone acetylation, which generally loosens the DNA structure in these future queens. This essentially opens up the DNA, allowing queen-specific genes to be expressed more easily. The worker bees, they don't get that royal jelly treatment, so they develop different epigenetic profiles that guide them towards their roles.
Wow, it's such a powerful demonstration. Environment, in this case, diet, fundamentally reprogramming the epigenome.
It really is. It shows how plastic development can be based on environmental input.
So if diet can do that, what about other environmental factors? Like you mentioned, population density. That brings us back to the locusts, doesn't it?
It does. Locusts are another, maybe even more dramatic example of this kind of plasticity. They can switch between being solitary and being gregarious, part of a swarm. And these two phases, they're not just a little different. They have significant differences in behavior, color, even their internal physiology.
And critically, these changes are reversible, right? It's not a one-way street.
Exactly. Reversible and largely triggered by environmental cues. The big one is crowding, population density.
So a locust living alone is practically a different animal than one caught up in a swarm, even though the genes are the same.
Absolutely. And the research, as the sources detail, is pointing more and more towards epigenetic mechanisms driving this phase change, this polyphenism. For instance, studies have found clear differences in DNA methylation levels when you compare solitary locusts and gregarious ones.
So again, the same DNA blueprint, but different chemical annotations depending on whether they're crowded or alone.
Precisely. And even more telling, perhaps, is that the genes encoding the DNA methyltransferase enzymes, remember, the ones that add the methyl tags,
Yeah.
particularly one called DNMT3, its activity is ramped up, upregulated, in the brains of gregarious locusts.
Okay, so the swarming locusts are making more of the machinery that adds these potentially silencing tags to their DNA.
Right. Which leads to the obvious question.
Could that be driving the swarming behavior? If they have more of these methylating enzymes, maybe that's pushing them towards those gregarious traits.
That's exactly what researchers tested. And it's pretty cool when they experimentally reduced the activity of this DNMT3 enzyme in locusts.
What happened?
Those locusts showed less of the typical gregarious behaviors. Specifically, the hyperactivity, that sort of frantic movement you see in a swarm, was reduced.
Huh. But did it affect everything? Like, were they still attracted to each other?
Interestingly, their attraction to other locusts seemed less affected. This suggests that DNA methylation, particularly controlled by DNMT3, might be especially important for the movement and excitability parts of swarming rather than the initial social attraction itself.
That's a really direct link, though. Manipulate the epigenetic machinery, change the swarming behavior.
It really is. It's compelling evidence. And further research, particularly on desert locusts, has found distinct DNA methylation patterns in the brain that correlate with this phase change, both short-term and long-term patterns.
Short term, too. How quickly?
Get this, even just 24 hours of crowding can induce measurable changes in neural DNA methylation. It's like the brain starts creating an epigenetic signature of becoming gregarious almost immediately.
Wow. So the brain is getting molecularly rewired epigenetically just by being around others.
That's what it looks like. And it's probably not just DNA methylation doing all the work.
Right. You mentioned other mechanisms earlier.
Exactly. The sources also point to suspected roles for histone-modifying enzymes and those small non-coding RNAs, the microRNAs, in locusts too. It suggests a complex sort of multi-layered epigenetic system controlling this whole phase shift.
A whole regulatory network.
Yeah. And there's even evidence, though it needs more research, suggesting that the parents' experience, like if they were crowded, might epigenetically prime their offspring to be more prone to gregariousness.
Transgenerational effects. So swarm parents might have swarm-ready babies.
Potentially, yes.
That's
A form of transgenerational epigenetic inheritance. It's a really fascinating area.
It absolutely is. Okay, so we've established this powerful role for epigenetics in locusts, driven largely by crowding. Now let's bring it back to our main, perhaps less charming subject, the flesh-eating screwworm fly. What kind of epigenetic factors might be involved there? It's not crowding, presumably, but the wound environment.
Right. Well, the New World screwworm, Cochleomya hominivora, is what we call an obligate parasite. Its larvae must have living flesh to develop. They can't survive on decaying matter.
Unlike some related flies.
Exactly. Like its close relative, the sheep blowfly, Lucilia cuprina. That one's a facultative parasite. It mostly feeds on decaying stuff, but can infest wounds if the opportunity arises. Comparing these two is really useful.
Okay.
Now, genomic studies have found genetic differences, for sure, that likely contribute to the screwworm's specialized, obligate, parasitic lifestyle. There's a genetic basis.
But genetics isn't the whole story, maybe.
It seems not. That 2021 study you mentioned, the one comparing gene expression across several fly species from pure scavengers to obligate parasites like the screwworm, that study was really revealing.
What did it find? What was different in the parasitic ones?
It found what they called convergent physiological adaptations in the parasitic larvae, meaning different parasitic species had independently evolved similar solutions. Specifically, they all showed increased activity upregulation of genes involved in dealing with iron, things like ferritin for iron storage, and also genes that help them tolerate low oxygen or hypoxic conditions.
Which makes perfect sense, right? A living wound.
Right.
Lots of blood means lots of iron, but the deeper tissues might be low oxygen.
Precisely. These parasitic larvae seem to be ramping up the production of proteins that help them thrive in that very specific, very challenging niche.
Okay, but how does epigenetics come in? That study measured gene expression, not the epigenetic marks themselves, right?
Correct. They didn't directly measure methylation or histone marks in that study. However, This pattern of differential gene expression genes being turned up or down, specifically in response to the wound environment, strongly suggests epigenetic regulation is involved. Think about the wound environment. It's warm, there are bacteria, there's the host's immune system fighting back. All of these could act as potent triggers for epigenetic changes in the larvae.
So the wound itself is like a complex signal, telling the larva's genes, okay, switch into parasite mode now, through epigenetic changes.
That's a very good way to put it. It's analogous perhaps to immune priming in insects. If an insect gets a small exposure to a pathogen, it can sometimes trigger epigenetic changes in its immune genes.
Making it better prepared if it encounters that pathogen again.
Exactly. So, similarly, the challenges of the wound, the bacteria, the host defenses, might induce epigenetic modifications in the screwworm larvae, turning up genes that help them, say, digest tissue, cope with bacteria, or evade the host's immune response.
Okay, that makes sense. So do we know if screwworms and their relatives actually have the necessary molecular tools, the epigenetic machinery, to do this?
They do. Now, DNA methylation seems to be generally less widespread or maybe used differently in flies, Diptera, compared to, say, bees and ants. But they definitely have other key epigenetic mechanisms. For instance, in the sheep blowfly, the facultative parasite researchers identified 5 different genes encoding histone deactylases, or HDACs.
HDACs, those are the ones that usually remove acetyl tags, leading to more compact DNA and gene silencing, right?
Generally, yes, and this is where it gets really interesting. There's an experiment where researchers treated sheep blowfly maggots with HDAC inhibitors, molecules that block the activity of these HDAC enzymes.
And what happened when they blocked them?
It significantly messed up the maggots' development. Their growth slowed right down, and it seriously hindered their ability to pupate, to transition to the next life stage.
Whoa. So blocking these epigenetic enzymes basically stops them in their tracks.
Pretty much. It strongly suggests that histone deacetylation, controlled by these HDACs, is absolutely crucial for normal larval development, and by extension, probably crucial for their ability to be successful parasites within a wound.
So maybe they need HDACs to turn off certain genes at the right time, or maybe to keep developmental genes tightly controlled?
Could be. Or conversely, they might need the opposite enzymes, histone acetyltransferases, HATs, to add acetyl groups and turn on genes needed for virulence, like enzymes, to break down tissue or suppress the host immune system. It's likely a dynamic balance. Adding and removing these histone tags, orchestrated in response to the wound environment, could be tuning the whole parasitic program.
So it's not just simple on-off, it's about fine-tuning the levels of gene activity. Very sophisticated.
It really seems to be. And it's not just histones. Remember those small non-coding RNAs?
Microarrays, yeah.
There's evidence for their role, too. Studies on a related species, the secondary screwworm, which usually arrives after the primary one, show that the levels of specific microRNAs change depending on the developmental stage. This suggests they're involved in regulating development and potentially responding to environmental cues, too, maybe like the specific nutrients available in their diet, whether it's decaying tissue or something else.
So different food sources could trigger changes in these microRNAs, which then tweak the expression of other genes involved in, say, metabolism or maybe even behavior.
That's the current thinking, yeah. These microRNAs could represent another layer of this epigenetic or epigenetic-like control, allowing the larvae to adapt quickly to different conditions.
Okay, so the picture is building. Environmental cues from the wound, likely triggering epigenetic changes. Histone modifications seem key. Maybe microRNAs, too, could be playing a really significant role in how screwworms do what they do. What about other flies that cause myiasis, this infestation of living tissue? Are there similar hints there?
There are, yes. Though, like you said, for many other species, the research is often more indirect or in earlier stages. Let's take the sheep blowfly again. That HDAC inhibitor study is pretty strong evidence that epigenetic regulation is critical for its development, which is fundamental to it causing fly strike in sheep.
Right.
And beyond that, there's speculation, but it's plausible speculation that epigenetic changes might be involved in how these flies develop resistance to insecticides.
Yeah, that's a big problem.
Huge problem. Or how they adapt to periods of nutritional stress. Maybe, just maybe, epigenetic shifts could even nudge a facultative parasite like the sheep blowfly towards becoming more parasitic if its usual food sources like carrion are scarce.
That makes a scary kind of sense, like a survival mechanism kicking in, epigenetically enabled.
It's a hypothesis worth exploring. Then there's another character, the hairy maggot blowfly. It has this really interesting switch.
What does it do?
Normally it feeds on carrion, like the sheep blowfly often does. But under stress, specifically starvation or overcrowding these maggots, can actually turn predatory. They become cannibalistic, preying on other maggots, including other species.
Wow, okay, that's a major behavioral shift from scavenger to predator.
It is. And when researchers looked at gene expression during this switch, they found changes in genes related to all sorts of things: cell signaling, stress response, maintaining water balance,
Yeah.
and interestingly, dopamine neurotransmission. Dopamine is a key regulator of behavior in many animals.
So the shift involves changes in brain chemistry pathways, too.
It seems so. And this rapid, profound change in behavior, triggered by stress, strongly hints at some kind of epigenetic switch being flipped, probably involving histone modifications, because those can alter gene expression relatively quickly.
So stress isn't just damaging, it can actually be a trigger signal for these quite dramatic adaptive behavioral changes mediated by epigenetics.
That certainly seems plausible based on the evidence. And then if you think about really specialized parasites like botflies, which live inside the host for a long time, or other flesh flies,
Right.
you can speculate further. How do they synchronize their development so perfectly with the host? How do they manage diapause, that period of arrested development, often timed to the host or seasons?
Right, that requires precise control.
Exactly. And how do they regulate the release of virulence factors, the enzymes, the immune-dodging molecules, potentially in response to signals from the host? Epigenetic mechanisms responsive to host cues seem like a really good candidate for managing all that complexity.
It really paints a picture of epigenetics as this crucial layer of flexibility, allowing these insects to adapt and thrive in these incredibly specialized and often pretty hostile parasitic niches.
I think that's a fair assessment.
So, looking ahead then, what are the big unanswered questions? Where does the research go from here?
Oh, there are lots of exciting directions. One really key area is exploring the potential interaction between hormones and epigenetics.
Hormones, like stress hormones.
Could be stress hormones from the host, maybe picked up by the larvae, or hormones produced by the larvae themselves in response to the wound environment. Could these hormones be the trigger or part of the signaling pathway that leads to the epigenetic changes driving parasitism?
Interesting connection. What else?
Another fascinating area is the microbiome, the bacteria in the wound, or even in the fly's own gut. Could these microbes be producing molecules that influence the fly larva's epigenome? There's growing evidence for microbiome epigenome crosstalk in other systems.
Wow, so the bacteria in the wound could be indirectly manipulating the fly's genes via epigenetics. That adds another layer of complexity.
It really does. And then, of course, there's that question of transgenerational effects we touched on with locusts.
Right. Could the parents' experiences prime the offspring?
Exactly. Could, say, a female fly that developed in a particularly challenging wound environment pass on epigenetic marks to her offspring that make them somehow better prepared for or maybe more inclined towards a parasitic lifestyle? It's speculative for flies, but definitely possible.
And thinking more practically, could understanding all this actually help us control these pests? They cause huge economic damage and suffering.
That's definitely a major goal and a really promising avenue. Remember that HDAC inhibitor study in sheep blowflies?
Yeah, stop their development.
Exactly. That shows that targeting these epigenetic control mechanisms could be a genuinely novel strategy for pest control. You can imagine developing specific inhibitors, maybe delivered as drugs, or perhaps using RNAi technology,
RNAi.
right? RNA interference. It's a way to specifically silence genes. You could potentially target the genes for key epigenetic enzymes in the pests. The idea would be to essentially flip their epigenetic switches in a way that makes them less virulent, disrupts their development, or even prevents them from establishing an infestation in the first place.
Using their own regulatory systems against them. That's clever.
It's an innovative approach, yeah, and it ties into broader trends. If you look at research on other parasites, malaria, schistosomes, even parasitoid wasps that lay eggs in other insects, epigenetics is emerging as critical for things like life cycle transitions, antigenic variation to evade immunity, and manipulating the host.
So it seems like a common theme in parasitism generally, using epigenetic flexibility to survive and exploit the host.
It certainly looks that way. There are likely common principles at play across very different host parasite systems.
Okay, so just to kind of wrap up this deep dive, it really feels like the big takeaway is that epigenetics offers this incredibly important flexible toolkit for insects like screwworms and locusts. It allows them to adapt, sometimes really rapidly, to environmental changes or challenges, leading to these, frankly, stunning shifts in behavior or development.
Agreed. And while we definitely need more direct epigenetic data for screwworms, actual maps of methylation or histone marks, The current evidence, especially the gene expression patterns and the comparative studies, strongly points towards epigenetic regulation being a significant factor in their specialized flesh-eating behavior.
Yeah. And having that locust example where the role of DNA methylation in swarming is much better established provides a really valuable comparison, a sort of proof of principle in insects.
Absolutely. It shows it can be done and how powerful it can be.
And the future research sounds fascinating. Those potential links between hormones, the microbiome, and the epigenome, exploring transgenerational effects.
Mm.
And especially the potential for developing new epigenome targeting pest control strategies.
Yeah, those are all really exciting frontiers. Lots
Mhm.
still to uncover.
It really makes you think, doesn't it, about how many behaviors we see in nature, things that seem so fixed and innate, might actually have these hidden layers of epigenetic flexibility underneath, allowing for much faster adaptation than we might assume just based on genetics alone. It certainly makes you look at the world a bit differently, encourages you to think about other examples where the environment might be profoundly shaping an organism's traits without rewriting its basic genetic code.
Well, thanks for digging into that with me. That was, well, both disturbing and incredibly fascinating.
My pleasure. It's definitely a topic that blends the gruesome with some really elegant molecular biology.